Everyone. So it's my pleasure to introduce the next session, session 4 on novel antimicrobial targets. And it's also my pleasure to introduce the moderator for this session, Jordi Vila, who's head of the Department of Clinical Microbiology at the Hospital Clinic in Barcelona, full professor of the School of Medicine of University of Barcelona, research professor in the Institute for Global Health of Barcelona, Spain, and also a member of the JPIAMR Scientific Advisory Board and Scientific Steering Committee for the workshop. So Dr. Vila, the floor is yours. Thank you very much.
Okay, thanks a lot, Mark, for this introduction. Good morning, everyone, and welcome to this session 4, entitled Novel Antimicrobial Targets. As you all know, when we talk about antimicrobial resistance, we can take into consideration two aspects. The first one is the emergence of the antimicrobial resistant bacteria, and the second one is the spread of this antimicrobial resistant bacteria. And when we say that, I say that because we have to take different actions according to if we want to stop the emergence or we want to stop the spread of multi-drug resistant bacteria. And these actions has to be taken from what I call the local level to the global level. Taking into consideration the local level as the hospital, is the first level is the hospital. In the hospital, we can run all the antimicrobial stewardship programs, all the hygiene criteria to control either the emergence and the spread of multi-drug resistant bacteria. So it's a small niche that we can control easily, but there is a flux, a continuum between the hospital and our community. And in this sense, the community is the second level. And in the community, there are four players that play an important role in trying to control the antimicrobial resistance, emergence, and dissemination. The players are the people living in the community, the pharmacists, the general practitioners, and also the veterinarians. Because as you know, when we talk about antimicrobial resistance, we talk about One Health. We talk about the interaction between the environment, the animals, and the humans. In this sense, there has been also described a list of different actions or activities to perform to control, again, the emergency and the dissemination of antimicrobial resistant bacteria. The third level will be the national level. Most of the countries in the world have developed the action program, antimicrobial resistant action plans to control the antimicrobial resistance. And the fourth level, the global level, will be an international level. I mean, the interaction, an activity done in one country can affect another country. We have continuously driving or transferring multi-drug resistant bacteria from one site to another one. So we need a integration of all these plans. In addition of that, of course, we need more antimicrobial drugs. In this sense, you can say, well, if you control very well the emergence and the spread of antimicrobial resistance, that's help. Of course, that helps. But for instance, there is a break in Europe between the north and the south of Europe. The Scandinavian countries has more control on antimicrobial resistance comparing with the Mediterranean countries. However, if we see the trend in the antimicrobial resistance, for instance, in ESBL producing Enterobacteriaceae in the Scandinavian countries, there is also an increase. The slope is not as pronounced as we have in the Mediterranean countries, but there is an increase. So in this sense, we desperately need new antibiotics, no? As you know, there is a plethora of different strategies to develop new antibiotics, phages, antibodies, new targets, peptides, blocking antivirulence factors, or blocking virulence factors, and also blocking the mechanism of resistance. So there are a plethora of different mechanisms or strategies to develop new antibiotics. And this is what we will cover in this session. There will be 5 interesting lectures in different areas, different strategies to develop new drugs. So with this, I just would like to introduce the first speaker. First speaker is Jean-Claude Sirat. He's a scientist working in the Institut Pasteur of Lille, and he will talk about the boost of innate immunity to treat bacterial pneumonia. Jean-Claude, it's your time.
Can you see my screen?
Yes.
Okay. So within the next 10 minutes, I will present you the work that we have conducted in the JPIAMR call and the project Abimune, so that aim to boost the innate immunity to treat pneumonia. So this work was conducted at the Center for Infection and Immunity of Lille at the Pasteur Institute. And the main, I want to acknowledge the main contributor, that is talented postdoc, Ferdinand Kazilag. This work has been conducted in coordination with 4 other teams, Charlotte Clough at the Freie University of Berlin, Tom van der Pol at the AMC in Amsterdam, Mustafa Sitar in Tours in France, this research center, and Martin Rumbo at the University of La Plata in Argentina. So the idea of the Abimune project was to develop alternative to therapies to antibiotic-resistant bacterial infection. And for this, we plan to repurpose antibiotics with non-antibiotic drugs, especially immunostimulatory molecules, and with the aim to treat lung bacterial infection. So our approach is a non-traditional antimicrobial approach that can be positioned as an adjunct to quench infection or to avoid treatment failure. So why targeting the innate immunity, especially the inducible innate immunity to treat infection? First, it's a natural system of antibacterial defense. Innate immunity is built-in and universal. It is independent of the individual, or I would say the pathological background of the individual. It's ubiquitous, so most tissue and body compartments can be activated by this system. The other interesting aspect is the kinetic of this response. It's immediately inducible within minutes, and it also self-controlled. So it regulates itself the switch off of the system. It also mobilizes multiple antibacterial immune effectors and immune cells, and both have a broad spectrum of antibacterial activity. And this is less likely that the bacteria develop resistance to such immune defenses. Most of the time, it's independent of antibiotic-resistant mechanism. And at the time we started this project, it was mainly envisaged in the prophylaxis context, so to avoid infection. And it's pretty well, I would say, documented on this side. So to target this inducible immunity, we focus on the toll-like receptors because we have expertise in the consortium that are receptors that are able to detect bacterial compounds. These are different bacterial compounds from various microbes, various bacteria, parasites. And we focus on the toll-like receptor 4, this blue molecule. And we used to induce this signaling, the monophospholipid. I will come back to this later. So what does toll-like receptor signaling? It induces a complex cascade of signaling that feeds in NF-kappa-B and also MAP kinase signaling, and this induces a transient activation of immunity. So this is the main picture. So what is the MPLA, so the molecule we use as the adjunct? It's a pharmaceutically approved biologic that targets TLR4. The name is monophospholipid. So it's a lipid with only one phosphate substitution, and it can be also substituted with 5, 6, or 7 acid molecules. So it's a mixture of active molecules. The one we are using is derived from Salmonella minnesota, lipopolysaccharide. It's immunostimulatory via TLR4. It has been well documented, and we further documented it. There is no toxicity compared to LPS. And interestingly, this molecule is used as an adjuvant in human vaccine, especially vaccine against papillomavirus and hepatitis B virus. As the trademark is AS04. To analyze the effect of this MPLA as an adjunct, we use a model. So we use Streptococcus pneumoniae. So it's a gram-positive bacteria. It's a principal cause of pneumonia, septicemia, and bacteremia and meningitis. It's a major cause of mortality worldwide, both in infants and in the elderly. It's one of the top 10 priority bugs for antibiotic resistance. And interestingly, the individuals that are deficient for innate immunity are usually of high susceptibility to this infection. So we set up different infection model, and the one I will present you in a few slides what the major take-home message of this study. So we use a model of infection in mice with respiratory infection using an antibiotic susceptible Streptococcus pneumoniae. 12 hours later, we proceed to different treatments, so a Merck treatment. Treatment with amoxicillin at 10 micrograms, that is a subcurative dose of amoxicillin in this model. That mimics what could happen, I would say, with antibiotic resistant strain using very high dose of amoxicillin. Amoxicillin was administered by the oral route. We use MPLA by intraperitoneal route at a dose of 50 micrograms. It was a unique dose. And we use a combination of this treatment, and then we measure the bacterial counts over time. So as you can see in the panel on the right, after infection, we see that the Merck treatment cannot enable the control of infection. There is a bacterial burden in the lung. Amoxicillin subcurative dose is doing poorly. MPLA is doing a little better, and amoxicillin plus MPLA is really increasing the clearance, or at least prevents the growth of bacteria very efficiently 36 hours post-treatment. It's the same in the spleen because this disease is giving us a rise to an invasive disease. We have the same picture regarding the survival. So here you see the Merck and Amox that are really poorly protected. MPLA is doing better. You can see that there are a certain amount of protection, and Amox plus MPLA is really increasing the survival. And beyond this, if we look at the 2 days post-infection, the histopathology of the lung, you can see that the Merck has a very bad histopathological score. Amox is doing a little bit better, but not significantly. MPLA is doing really better, and the combination is really increasing the quality of the lung architecture, meaning that there is a good protection against pneumonia. We have also combined that with modeling. So we have used PK analysis and PD analysis, and we combine that with nonlinear mixed effect modeling and time to event analysis. And on the top of the pharmacokinetic interaction, we have defined that the pharmacodynamic effect of a combined treatment was superior. And we are still continuing this pharmacometrics analysis to go to the clinical setting. So just as a first conclusion and the next step, so we can conclude that boosting innate immunity as a general concept is a strategy of interest to improve the outcome of antibiotherapy and to decrease the dose of antibiotic. It still remains to define what is the immune mechanism. It's a hit and run mechanism that is involved in the clearance of bacteria and resolution of infection and inflammation. We have already performed the Macro Rna analysis to have some idea about this. And finally, I think this is perhaps the next step. It will be to attract some other partners to go beyond our proof of concept using pharmaceutical grade molecule and relevant model with gram-negative bacteria. So we wanted to go further in this project. So one of our ideas was to compartmentalize the innate immunity because this innate immunity is ubiquitous. So we wanted to bring it back to a very focused targeting on the airway. So we decided to do aerosol delivery to promote airway signaling. Since MPLA is a poor activator by this route, we focus on the recombinant flagellin from Salmonella, that is an activator of TLR5, and that we started to work on in the JPIAMR project. So to go further, we extended our network of academic and industrial partners and moved from preclinical study to clinical study. We applied for an EU project that was funded, and the idea is to go to phase 1 clinical trial by nebulization to do a first-in-man study. And to set up the capabilities for the first-in-man clinical trial, create the environment to analyze the mode of action of a general immunostimulator and assess the ecosystem that is regulatory, social, and economic. And this is how we say the bigger picture, and I will stop here, and I will be happy to take any questions.
Well, thank you very much, Jean-Claude. You stay on time. Perfect. Thank you. Thanks a lot. So let's move to the second speaker. It's Professor Morten Kos. He's a professor, associate professor in microbiology at the Norwegian University of Life Science in Norway. And he will talk about fighting antimicrobial resistant infections by high throughput discovery of biofilm disrupting agents and mechanisms. This is inside of a project called Disrupt. Morten?
Okay, thank you. Do you hear and see my slide?
Yes, perfect.
Thank you. So again, thank you for the opportunity to present an overview of our project here at this workshop. So I'm coordinating a JPIAMR funded project, which we call Disrupt, which is currently ongoing. And the focus of this project is to find novel strategies, targets, and tools to be able to fight biofilms. So this project is really a discovery project where we want to develop new research tools and use high throughput approaches to discover agents and mechanisms that can be used against biofilm forming pathogens. So the consortium team consists of 4 research groups located in Norway, Germany, and Switzerland. So it's our group at the Norwegian University of Life Sciences. And there are groups of Nasos Typas and Christoph Merten at EMBL and Jan Wilhelm Feining in Lausanne in Switzerland. So our project focuses on biofilms. So as you all know, biofilms are communities of microorganisms which are formed on different surfaces. So biofilms consist of cells, often in different metabolic states, and they are enclosed in some kind of extracellular matrix composed of either polysaccharides, extracellular DNA, or proteins. These biofilms are associated with infections, also those caused by AMR pathogens, both gram-negative and gram-positive pathogens, including, for example, infections such as cystic fibrosis, chronic wound infections, and infections related to medical devices and catheters. So biofilms are extra problematic because they're not only a problem in themselves, they're also the bacteria residing in these settings. They are extra resilient to treatments and also resilient to our immune system. So this is both because the matrix protects them, but also because cells residing in the biofilms may be dormant or in state of the persister cells. And within these biofilms, there may also horizontal gene transfer, so transfer of resistance genes may occur. So one strategy to kind of fight this would then be to find ways to inhibit biofilm formation or to somehow disrupt it, making them again more sensitive to antibiotics and be able to, or allowing the immune system to clear the infection. So for our project, we are focused on some bacteria which are relevant in an AMR perspective and which also lines up with our expertise. So the first aim of our project is to find and provide a systematic overview of novel anti-biofilm targets in this bacteria. So we're also working with uropathogenic E. coli, Staphylococcus aureus, Pseudomonas aeruginosa, and Streptococcus pneumonia. And would like to find novel ways to inhibit and disrupt these biofilms formed by these bacteria. So the second important aim in our project is also related to the development of genetic or new genetic tools and resources. So To allow for a systematic screening of targets and mechanisms, we are of course dependent on genome-wide approaches, as we heard also in this workshop, many use. So this allows for the study of all the genes in the organism at the same time. And I think furthermore, both for our project and also for research on AMR in the community, it's always critical to understand the molecular mechanisms which are at play if you identify a new compound. So, and how different agents affect the cell. So to help this, in this project, we are developing some genome-wide mutant collections, which we believe both in our project and also for others in the future will greatly speed up this process of identifying mechanisms. So our project is, we have divided it in 3 parts. The first part is regards to development of new tools. So what we are doing here, we are creating ordered transposal knockout collections. Using transposal mutagenesis, we are making in our selected strains, we make pooled genetic or transposon libraries. So this will then be in the same culture, a lot of mutants. Using transposal sequencing, this will, first of all, it will allow us to define the essential genes, which can be used as direct targets. It's also defined that, which of course also has been done before. But then we use these pooled collections with the help of robotics to array them into ordered collections. So we use so-called combinatorial pooling with the help from colony picking robots and pipetting robots, and make this ordered collection. So the result would be multiple plates consisting of ordered mutant collections. So the potential issue with, or potential problem with transposal mutant collections is that they will only allow studies of essential genes, non-essential genes, because knockouts of essential genes would not survive. So a solution to this is that we also create knockdown libraries of the same strain. So for this, we use CRISPR interference, which is a transcriptional knockdown strategy. We create first pooled libraries of these inducible CRISPR with these inducible CRISPR systems, and in the next step, again using robotics to order them into ordered collections, thus allowing also studies of essential genes in this process. These mutant collections are then, we want to use them in our next step, or we are using them in our next step, which is to identify or systematically characterize which genes that are essential for biofilm formation. So we want to do this in a high throughput manner. So to allow this, we are using kind of simple biofilm assays. We use crystal violet assays for liquid biofilms, and we use structured macrocolonies to look at biofilms formed on agars. So these forms of biofilms are not necessarily representative. I mean, biofilms depends a lot on different environmental conditions, but this were the methods we chose for our high throughput assays. So basically, what we do then is to assay all our mutants to see how they're affected in biofilm phenotype, and look for a correlation also between these two methods and also between strains. So finally, we also wish to find new compounds and new targets. For this, we are using 2 approaches. We are screening different chemical libraries. Again, we use a high throughput setup and robots to screen chemical libraries to find compounds that affect or reduces biofilm formation. Secondly, in the group of Christoph Merten, they are using, or they are screening for antimicrobial antibodies. With this approach, antibody-producing cells from immunized mice are encased in droplets together with bacteria, and then based on fluorescence sorting, we're able to identify antibodies that are antimicrobial and then develop those further. So this is an ongoing project. So the progress so far is that we are developing the tools, and we are already having some candidates that we are following up on. So the final aim, or the aim of this project, would be to come up with or have some systematic knowledge about the biofilm formation in these strains, and also come up with some novel mechanisms and for some antibiofilm agents. Okay, then thank you.
Okay, thank you, Morten. You have to stick to the time, so thanks a lot. So let's move to the third speaker. We will have questions at the end of the 5 speakers talk. The third speaker is Vipul Panchal. He's working in the University of Bergen. He's a scientist there. And he will talk about the antibiotic rosoflavin targets, flavoproteins, and FMN riboswitches. This is inside of the project entitled Explore.
I hope I am audible. Hello?
Yeah.
Yeah, okay. So I think there is some mistake. So the title of the project that I'm working on is Exploring the TPP riboswitch as a novel target for antibiotics. It's also a part of Explore joint program initiative. So yeah, so in the context of typical bacterial transcriptome, if we consider all existing antibacterials, they either target this protein coding region or ribosomal RNA region. Now, riboswitch here are basically non-coding RNA element that resides within the 5' untranslated region of the transcript. And therefore, targeting riboswitch allows us a unique opportunity to explore this relatively untouched non-coding RNA fraction of bacterial transcriptome as a novel antibacterial landscape. Now, riboswitch elements binds to the target, the cognate ligands, and to regulate the expression of genes involving their own metabolism. Now, typically, riboswitch elements has 2 distinct domains, and the aptamer domain that binds to the ligand, and expression platform then that in response to this interaction either turns on or turns off the expression of the downstream gene. In most of the cases, it turns off the expression of the genes. Now, in case of TPP riboswitch, it binds to thymine pyrophosphate. So TPP is basically a ligand, which means TPP riboswitch in response to thymine pyrophosphate turns off the expression of the genes involved in TPP biosynthesis. Now, there are few more criteria that qualifies TPP riboswitch as a most suitable antibacterial drug target. And in fact, in our recent review, we discussed few selective riboswitch elements for their potential as an antibacterial drug target based on several criteria. So coming back to TPP riboswitch, it is almost exclusive to the bacteria, and most importantly, they are absent in humans, which means it allows us to achieve high degree of selectivity. Now, because it binds to TPP, naturally they have sophisticated druggable binding sites, and therefore it is possible to run intentional drug discovery programs. Now, since TPP is an essential cofactor, TPP riboswitch mediated regulation is essential for the bacteria. Further, they are highly prevalent among bacteria. So TPP riboswitch is present in 46 human pathogens, and therefore it serves as an antibacterial for antibacterial target, pan-spectrum antibacterial target. Now, aptamer domain of TPP riboswitch is highly conserved. So here we are looking at the sequence representation of sequence alignment from thousands of bacteria. And as you can see, it is highly conserved. Now, proof of principle for TPP riboswitch as an antibacterial drug target already exists in the form of naturally occurring antibacterial compound pyridine. Now, this compound targets TPP riboswitch as well as proteins involved in the TPP biosynthesis. And because in humans, the protein counterpart already exists, this compound could not qualify as an antibacterial drug. Now, as soon as TPP riboswitch was discovered, Chen and al. Undertook the fragment-based screening approach and identified fragments that binds to TPP riboswitch. However, since then, no further efforts have been undertaken to chemically elaborate them in order to identify potent compounds. Alternatively, Yunse et al. Looked for the structural analogs of its cognate ligand, that is thymine pyrophosphate, and identified trizolethylamine as one such compound. But because it's a structural analog, it's likely that it has off target. So in conclusion, there is a lack of comprehensive effort to explore full potential of TPP riboswitch as a drug target. So in this consortium agreement, in collaboration with experts from the different disciplines, Professor Mack from Germany, Professor Lefontaine from University of Sherbrooke, Dr. Shedlark and Dr. Bartonuk from Czech Open Screen, Professor from Norway, and Professor Gins from Dr. Gins from Latvia. We are focusing on exploring the TPP riboswitch against escaped group of pathogens and as pneumonia. So one of the main objective is to develop high throughput screening assay against this riboswitch, because as such against the RNA targets, there is no significant development in this aspect. This will be followed by the high throughput screening against a large library of compounds. And this includes compounds from Czech Open Screen, EU Open Screen, and in-house compounds. This will be followed by improvement of affinity of validated hits through chemical elaboration. The second round of compounds will be subjected to antimicrobial activity profiling against escape group of pathogens and S. pneumonia. And finally, the most potent compounds will be studied for their mode of action. So in this aspect, I started with exploring the potential TPP riboswitch from escape group of pathogens and S. pneumonia. And this sequence were then filtered through certain criteria to end up with having sequences that has high probability of having riboswitch activity. Now, since the assay that we are interested in requires TPP riboswitch to have transcriptional means of regulation, I started working with one such sequence from S. aureus. So, of course, the first step is to first validate the riboswitch activity and means of regulation of this sequence from S. aureus. So, I perform in vitro transcription in presence and absence of ligand. So, here in the chromatogram, in the first lane, when there is no ligand, the percentage of read-through transcript, that is the full-length transcript, is 67%. Now, in the presence of TPP, which is a cognate ligand, this percentage of read-through transcript reduces to 36%. And now, if we use non-binder instead of cognate ligand, this could be restored to 62%. So this confirms that the sequence we selected from S. aureus has transcriptional means of regulation, and it's a riboswitch. So we further confirm this by calculating the by estimating the half maximal transcription termination value, which is 0.6 micromolar. Now, next step was to develop the assay, and this so we were interested in developing this dual reporter assay, originally developed by one of our collaborators from University of Sherbrooke. So as name suggests, the assay requires to relies on two molecular probes, which are basically oligonucleotide sequences labeled at 5' end with quencher and at 3' end with fluorophore. And because of this hairpin loop conformation, the quencher resides within the proximity of fluorophore. And so by default, there is a quenching of fluorescence. Now the assay requires us to perform in vitro transcription in presence of these dual molecular beacons under varying condition of ligand. So basically, we are recording the status of the transcript in absence or presence of the ligand. So just to illustrate further, in the absence of ligand, since there is no terminator stem in the expression platform of the ribose switch, the full-length transcript is synthesized. However, in the presence of ligand, because there is a terminator stem in the expression platform of the ribose switch, only truncated version is synthesized. Now, since one of these molecular beacon is complementary to the sequence at the 5' end, it gives fluorescence regardless of the presence or absence of the ligand. Now, on contrary, 3' another beacon is complementary to the sequence at the 3' end, and therefore it gives fluorescence only if there is no interaction between aptamer and ligand. So based on this principle, we plot a graph of fluorescence intensity from 3' beacon normalized with a signal from 5' beacon over time. And as shown in this graph, there is an incredible difference in the ribose switch activity between two conditions, that is without ligand and with ligand. And indeed, for Asorius thyri ribose switch, we could recapitulate the same difference in the ribose switch activity. That is, without TPP, there is a high possible ribose switch activity, whereas in presence, it is negligible or very low. So the next step is to miniaturize this assay, and this is what currently we are involved in collaboration with Dr. Sedlak at CIHR. So once we have this reproducibility and quality satisfied, we will be soon doing the compound libraries, high throughput compound screening. So meanwhile, Anna from Professor Max lab, she has developed the beta-galactosidase based reporter system in order to do this antibacterial activity profiling as well as the mode of action studies. So this construct requires LEGZ gene to be in fusion with TPP ribose switch, and this construct is then transformed to E. coli, particular E. coli strain, which is absorptrophic to the thymine. And here, and then she has looked at the ribose switch activity for different ribose switch from different organisms, that is here E. coli and B. ceteris. And as here the activity is defined in terms of beta-galactosidase activity. And as we can see here, only in the case where there is a construct with TPP ribose switch, there is a response to thymine ligand, which means in absence of thymine, there is no signal, and with increasing concentration of thymine, the activity is reducing. So it's currently testing the ribose switch sequences from escape group of pathogens as well as pneumonia, and she has already screened a few of the compounds with this activity. So that's all we have at the moment. So these are our future perspective. That is, the immediate objective is to do high throughput screening as soon as we are satisfied with the performance of the assay. And this is what our consortium group looks like. And I would like to thank our various funding agencies who supported this project, and yes, of course, JPIAMR and UKRI for inviting us for this talk. Thanks.
Okay, thanks a lot, Vipul. Yeah, let's move to the next speaker, who is Dr. Javier Sancho. Dr. Sancho's a professor in the School of Science in the University of Zaragoza in Spain. And he will talk about the novel antimicrobials specific against Helicobacter pylori in a project called FLAB4AMR. Javier?
Thank you, Jordi. Can you hear me?
Yes, perfect.
Okay, I will present novel antimicrobials that were initially developed in my lab at the University of Zaragoza, and that now are being developed in the frame of the FLAB4AMR project in collaboration with two French groups led by Elie Toiti at the Institut Pasteur, Alain Bousquet-Melou at the École Nationale Vétérinaire de Toulouse, and a German group led by Ulrich Scheible at the Research Center Boston. Helicobacter pylori, as you know, is a gram-negative proteobacteria that infects around 50% of the human population and causes peptic ulcers, stomach cancer, mold lymphoma, and therefore it is advised to be eradicated from patients, at least in cases of dysplasia and mold lymphoma. The the eradication therapy that has been commonly used is called triple therapy because it relies on the combination of 2 antimicrobials plus a PPI, and it fails already due to resistance in 30% of cases. So now there is a quadruple therapy consisting in the administration of 4 different chemicals that is failing in around 10% in as much as in what I have been reading recently. Our approach towards developing specific antimicrobials for this bacterium has been target oriented. We have focused on a protein that is the target of our discovery program. The name of the protein is Flavoxin, and this is an electron transfer protein that is present in many bacteria, but not in humans. It plays roles in a variety of important biochemical reactions. In some bacteria, it is an essential protein. In other bacteria, it is not, so it has to be established in a case-by-case basis. For Helicobacter pylori, we know that flavoxin is essential because it mediates in the oxidative decarboxylation of pyruvate to produce NADPH. So our assay was initially focused in discovering inhibitors of this reaction that we could identify because they failed to allow the production of NADPH in a spectroscopic assay. Our global approach has been screening chemical library against the target, and once we have found hits, we have done a couple of rounds of hit-to-lead work or improvement, accompanied with testing in animal models, precisely in a mice model of Helicobacter infection. We began with 10,000 chemicals commercial library and purified flavoxin, and we identified 29 binders. We did the identification of the inhibitors in a 2-step fashion. We first identified binders, which for us is easy using biophysics, and then we identified among them the 4 inhibitors using the previous spectroscopic assay that I mentioned. 4 were inhibitors, 3 were bactericidals, and in our first round of improvement, we characterized 150 analogs. Then we synthesized new analogs in a more focused manner, and in the end, we decided that we have one lead, which is represented by this compound, which is 7-nitrobenzoxadiathol. This is the lead of our development. Some of the characteristics of these compounds is that they have not shown resistances yet, which is not surprising because they have not been using, but we have tested how prone they were to developing resistances. So far, we didn't succeed in finding resistances or in creating resistances, while at the same time we were obtaining metronidazole resistant strains. On the other hand, we have shown that these flavoxin inhibitors are not substrates of the major efflux pumps, which I think is a promising result. Presumably, they are less damaging to microbiota. We have to test this because according to our data, the lead compound, compound 4, is quite specific for Helicobacter pylori, although it also shows efficacy for Campylobacter jejuni. Still, this compound kills several gram-positive bacteria, but most of the derivatives are extremely specific for Helicobacter pylori while retaining the activity. We have tested these inhibitors against clinical isolates found in Zaragoza hospitals. These 6 clinical isolates resistance to clarithromycin, metronidazole, or rifampicin, and again, some of the derivatives show fairly low mix against these bacteria, these clinical isolates. So we hope that they could be a good replacement for treating infections when resistances have arisen for this bacteria. We have done some toxicity studies in mice. The lead is not toxic except at very high concentration. At the concentration is shown in the screen, it is not toxic at all in mice, and the derivatives are not even toxic at twice as much that dose. We have done also some efficacy testing in vivo in the laboratory of Eliet-Touati at Paris, and many of these derivatives of compound 4 reduce the bacterial load in infected mice, and more importantly, they have been able to eradicate the infection in some mice, not in 100%. The best results have achieved eradication in 50% of the cases. But I would like to emphasize that we have been using a highly non-optimized administration protocol consisting in a single daily dose for 7 days and combined with any other antimicrobial or PPI. I mean, these results are obtained using just one derivative without no PPI. So I believe there is room for improvement. And now in the frame of the project, the FLAP4AMR project, we have found out some synergism between some of the derivatives and one specific PPI, not any of them. The specificity, we have confirmed that these compounds are quite specific for Helicobacter, but not only Helicobacter pylori, but also other Helicobacter species of human and veterinary interest. The effect on microbiota is being investigated in Borstel, also the pharmacological properties. We have synthesized more water-soluble derivatives which have higher bioavailabilities and metabolic stabilities, and they are ready for efficacy testing. The dosage regime is being investigated in Toulouse, and we hope to soon being able to perform efficacy tests of the novel, more water-soluble derivatives in combination with the PPI, for which we have found synergism. So we have advanced quite a lot in the preclinical, but we are far from the end. So we would be welcoming collaboration from business partners that could be interested in finalizing these preclinical studies. So we are open for proposals. Thank you very much.
Okay, thanks a lot. So let's move to the last speaker, Fadi Soukari. He will talk about, well, he's a scientist in the National Biofilm Innovation Center. He will talk about the sensitizing Pseudomonas aeruginosa biofilms to antibiotics and reducing virulence through the novel target inhibition. This is a project called Symbioter. Fadi?
Yeah. Pleasure. So, as been highlighted already anytime during this workshop, I mean, there's only few novel antibiotic over the last 20 years, novel chemical entities, and precisely there are only 4 so far. We followed the antivirus approach, which is becoming very popular over the last 2 decades, and mainly because it addresses unmet clinical needs, basically trying to reduce the resistance due to reduce selective pressure on bacteria, because this type of medication or drugs or candidate, they don't kill bacteria, they just disarm the virulence. The other unmet clinical need, which is preserving the natural flora. So basically, our study pathogen was Pseudomonas aeruginosa, and as you all know, gram-negative pathogen manifested in various infections from lung to cystic fibrosis to burns. It holds an armory of virulence factors, actually wide armories, from ability to form biofilms, immune invasion, antibiotic resistant cytotoxins such as pyocyanin, iron scavenging. So it's really nasty bacteria when it comes to infection. Specifically, we targeted chrome sensing, and this is basically the way that these kind of bacteria and other similar bacteria communicate with each other through chemical signals. So as you imagine, there is a receptor for this chemical signal in each bacterial cell and there is a synthase which is responsible for the synthesis. And this way of communication is basically to harmonize population, whether in planktonic or biofilm lifestyles, and harmonize gene expression, toxin production. So normally, when populations start to grow, the threshold or the concentration of this signal increases to reach to a threshold where the disease or infection starts. So it's really linked to the disease model itself. So in Pseudomonas, there's 3 quorum sensing systems. They're all interconnected between each other. There's the LAS and RHL systems, and there's the PQS. So our study was more focused on PQS system, which is based on chemical signal derived from alkyl quinolone scaffolds. And the receptor protein for this signal was PQSR, which was the main target in SAMRC program. Obviously, this system collaborates many variance factors from cytotoxin to biofilm to efflux pump. So it was quite validated drug target. So I just want to go quickly over the regulation. So the synthase in PQS is an operon with few genes on it, PQS A, B, C, D, and E. And this is actually drive other enzyme to make the main alkyl quinolone signal or the quorum sensing PQS signal. And this is drive virulence. Importantly, for this to work, as soon as the quorum sensing signal bind to receptor, it induces the synthesis of this chemical and provide a positive feedback loop until the concentration reach a certain threshold. So the hypothesis in SAMRC was if we block the binding and the reception of these signals, we could block the synthesis, and then we could disrupt this interaction and then disrupt the virulence, obviously. So as a general drug discovery, all drug discovery targeting quorum sensing been like the focus being on 3 areas. Signal molecules, some people try to sequester this through antibodies, enzyme, degrade them. Reception, which is blocking receptors, and this is what we did in SAMRC, and then the synthesis, target one of the biosynthetic enzyme. So this SAMRC program finished in 2018. It was funded by JPIAMR, and it was across 4 universities: University of Nottingham in the UK, University of Copenhagen, University Laval in Canada, and Uppsala University in Sweden. So we took over the medicine chemistry and microbiology. In Copenhagen, they did the antisense, peptide, nucleic acid. Laval is the animal study, and drug delivery in Uppsala. So it was a complete drug discovery program. So in Nottingham, basically this was my work. Just gonna go through it really quickly. As many projects presented already, we followed in silico screening, virtual screening for our in-house drug compound library, which we have. It's around 80,000 drug diverse compounds, and we did obviously like validation for the result, and we obtained around 4 hits. The screening was done at micromolar, and we disregard any inhibition less than 50%. One of the conditions is no effect on the growth, because that will render the compound non-antivirulence at all. So this is some of the hits that we obtained. Various PhD students took over other hits. I'm just gonna focus on the first hit. So we did some hit optimization. We managed to improve the potency from 3 micromolar to 67 nanomolar. We obtained crystal structure, and this was one of the most potent PQSR inhibitor reported in literature at that time. We did structural biology, binding assay with ITC. We did also phenotypic analysis. We measured the expression of the alkyl quinolone signal, which was hugely reduced in the presence of this inhibitor, as well as other toxin governed by PQS system, which is biocyanine. We tested also several clinical isolates, and that was mainly for pyroxene production. And we noticed there's still huge reduction, although it's variable. And this is highlight really the importance of studying clinical isolate in each project. We then moved into biofilm study. So these compounds have like marginal effect on their own, but they have to be adjunctive or a potentiator for antibiotics. So we did a biofilm study with tobramycin, and we can see the effect clear with and without the inhibitor. At 6 hours with the inhibitor, almost complete eradication of biofilms. We did cytotoxicity study, and they were like the concentration we are using, they were really non-toxic. And eventually we managed to perform some in vivo infection model, lung infection model in mice, and that was on one of the early candidates. Unfortunately, we didn't see a significant reduction in the infection, and that was mainly due, as the PK study showed, the short half-life of the compounds in one case, and we had to adjust the dosing and delivery. But unfortunately, by that time, the project finished, and we couldn't pursue any further in vivo work. On the other side, I wanted to also show what our partner in Copenhagen were doing. So basically, they were making peptide nucleic acid, and they were targeting not the receptor PKSR, but they were targeting one of the synthesis protein. So basically, these antisense bind to RNA for the translation of PKSA gene, which is the first gene in the sequence, and basically you have less alkyl-quinoline expressed or none. This is the same way of small molecule. This was validated with phenotypic assay in pycyanine, as well as effect on growth and biofilm in conjunction with antibiotic. So then we looked at the synergism. There's synergetic effect between one of these PKSR potent inhibitor and the antisense, and we managed actually to show there's a huge reduction when combined together in the IC50, and we managed to get a whole cell based assay with only 14 nanomolar. So this was almost the end of the scientific output. We had published so many papers out of this, and we're still actually writing around another couple of papers. It was a really successful program, and if we have the chance to take some of the stuff that we achieved further, we would like to improve the lead, obviously, enhance the half-life. We would like to optimize the drug delivery, formulation, and the route. And we would like to see the effect of biofilm of this inhibitor on biofilm, a preformed biofilm or polymicrobial models, which is really not really highlighted in the literature. And eventually, we want to study the effect of this inhibitor on blocking the immunomodulatory effect of other colon sensing molecules.
Paddy, please, can you finish?
Yeah, and that's all for me. So, so thank you very much for the funders and for all the people who assisted with this project.
Great. Please, can all the speakers show themselves?
Yeah.
Well, let's start. There are several questions in the chat. Let me start with this one. It's for you, Fadi. It's have you determined the internal concentration of the inhibitor necessary for antibacterial action?
So we always did wholesale based assay, and that basically we had no problem with efflux of inhibitor. So basically all the IC50 were done in wholesale assay. So as far as we know, there was no leakage of inhibitors outside the cell or efflux. So I'm assuming that in our case it was the same as our IC50.
Okay. No one for for Javier. You have used high throughput screening to identify inhibitors of flavoxin. Nevertheless, historically, this approach was not successful for identifying gram-negative antibiotics despite potent hits due to the inability of hits to penetrate the outer membrane core. What strategy have you used to impart this property to your binding hits against Helicobacter pylori?
We have followed no strategy at all, but we have worked with a well-defined target. We have identified binders, then inhibitors in vitro, and then we have tested them, and some of them have worked. So I don't think we should have any prejudice against high throughput or any other kind of throughput or low throughput screening, because they are just chemicals. Some will work and some others not. And the best we can do is to try and improve the defects they may have at some stage. So just try.
Yeah. Okay, there is another question probably for all, with exception perhaps of Jean-Claude. It's about the selection of resistant mutants. None of you said this question have shown this study about the selection of resistant mutants of your compounds or your drugs. Are you planning to do that? What is your idea on that? Who want to answer?
So I guess, yes, we would like to do that. So I mean, that's what the aim of the project, at least in my case, is. But so while we are considering the targets, it's also important to understand that the sequence conservation and whether they target multiple components within the pathway or multiple pathway or not. And that's how, so the targets that interferes with multiple pathway or multiple components within the pathway and have sequence conservation across the bacteria, they are the most suitable against this multi-drug resistant bacteria. At least it seems that way.
Okay, another one for Javier. Some of your clinical strains are naturally resistant to your flavoxin inhibitors. Do you know why and how to overcome natural resistance?
No, actually it's 1 out of 6, which I'm very happy that it's a low number. But at present, I don't know why this one is resistant.
Yeah. Okay, so no more questions. Well, I have several questions. Let me just check. Jean-Claude, do you think that using this strategy, you will have the same result with an gram-negative bacteria instead of using Streptococcus pneumoniae? Or you will have a more potent, perhaps, inhibition due to the fact that you are using lipid activation, flagellin activation, or?
Yes, it's a very interesting question. In the new consortium we have in the EU project, we'll address this issue with Klebsiella pneumoniae and Pseudomonas aeruginosa caused pneumonia. So I will have the answer perhaps in a few months or years.
Okay. and there is some papers showing that the intestinal microbiota can play a role in priming some macrophages of the immune system of at least in mouse. Do you think that the intestinal microbiota can play any role in this kind of studies that you are performing?
Yeah, it's there are several studies that show that if you have a dysbiosis, you have a change in the immune response, both innate and adaptive. So yes, for sure, if you modify it, you will have an impact for sure.
Great. Okay, we have more time. Let's see. So Morten, you talking about biofilm, that you looking for biofilm inhibitors. I see two scenarios here. No, I see an inhibitor to stop the formation of the biofilm, and another scenario who is the trying to destroy the biofilm. Do you think that could be two different inhibitors? Because when you have already the biofilm mature, probably the first steps has already been developed, of course, and then you have to look for two different drugs. How do you want to manage that?
Well, absolutely. This would be two different approaches. So for the so we are trying to do stuff in a bit like high throughput way. So in these setups, it's easier to like measuring the forming or not forming biofilm setup. But we are also trying to with already established biofilms to find agents that can kind of disrupt that. And I mean this could be molecules or, for instance, enzymes that degrade the matrix components, etc. So, but I mean, well, we are trying, but it's like from our we use our experimental approach. It's easier to find anti agents against those that form the biofilms.
Yeah, and you have selected 4 different bacteria.
Yeah.
But I miss Staphylococcus epidermidis.
Yeah.
I'm telling you that because you know it's causing an important cause of infection in prosthesis.
Yeah, yeah.
And if you are lucky enough and you have an inhibitor of the formation of biofilm in Staph epidermidis, and you can cover the prosthesis with this compound, then you stop the production of the biofilm, which is an important factor in infections caused by epidermidis. No?
Yeah, no, I agree, and also I think part of our project is also a bit like methods development. So if we find like a strategy that we could also later apply to the different pathogens. So the reason for choosing Staph aureus was that, well, also this is what we had most knowledge of
Sure.
and could work with. Yeah.
Sure. For Bipul, this is a question. It's based on that I have not so much knowledge on Arable Switch. You said that there is not an equivalent molecule in human cell, isn't it?
Yes.
Yes. And what is the difference between a macro RNA and a Arable Switch? Are totally different because macro RNAs are also involved in the regulation of some genes. So there is a lot of difference between both.
Yes, yes. So microRNA basically they anneal with the target sequence. So it's basically oligonucleotide based regulation. But in case of Arable switches, it is the small molecule or molecule based regulation. So as I mentioned, it binds to the aptamer domain, basically where there is a recognition determinant, and there is a regulation domain that basically either sequesters the ribosomal binding site or generates the terminator stem. That's how it regulates at transcriptional level.
Okay.
So what I did not mention during my talk is that specifically in case of TPP Arable switch, it regulates the multiple component of TPP biosynthesis, and that's why you can, by targeting TPP Arable switch, you can actually target multiple components within the pathway. So less likely to have resistance, at least on theory.
And what is specific Arable switch? If you find a Arable switch, I mean, inhibitor for Staph aureus Arable switch, that can be used in E. coli or in another bacteria, or is it very specific?
Yes. Yeah, they are. So as I think I showed in my slide too, that the aptamer domain, which basically recognizes the ligand, they are highly conserved across bacteria. So the conservation goes more than 70–80%.
Okay.
Yeah.
Okay. Let me see if there is another question in the. Is the CRISPR knockout collection of Staph aureus are available now?
It's not yet, but it will be in a little while. Yeah.
Okay. And I have a question for Javier. Javier, have you tested? You know that some groups said that the chronicity of the infections caused by Helicobacter pylori is related to biofilm formation. Have you tested your compound in a biofilm forming Helicobacter?
Not yet. Our German colleagues have expertise in biofilms, and they will test the compounds eventually. But so far, we don't have any results indicating that they act at that level. But we plan to have some tests.
Great. What else? There's another one for Javier. The lack of selection of resistance to your compound might indicate it has a multiple targets. Have you evaluated that?
No, we suspect that there may be an additional target, and we are now doing some transcriptomic and proteomic analysis.
Mm-hmm.
We think that the main target is flavoxin, but in some cases, we have suspected that there could be a more general effect of some of the compounds. So we suspect a possible additional target that we will eventually find out.
Okay, so.
There's some questions, Jordi, in the open questions. Do you want me to read them out? Would that?
Yes, please. Yes, please.
So there's one directed that Jean-Claude Sierra has said that he'd like to answer, which is, does MPLA induce the tolerance or allergy as LPS?
We have not tested that, but you know, MPLA is a very is a molecule that is highly attenuated, so it makes less, I would say, pro-inflammatory response than LPS, hopefully. Yeah? But yes, it has to be tested. Perhaps there is a tolerance effect.
There's one to Javier, which is, what is the effect of pH on your compound?
None. They are acid resistant, if the question is directed to that.
And then in the host environment, how can we make sure that the bacteria would maintain the Arable Switch structure?
How do we maintain? So I guess it's based on the homeostatic level of the ligands. So, but the aptamer domain seems to be remain conserved. So I don't think that is the concern in most cases. If there is a ligand will bind to the Arable Switch, and that will determine the fate of following transcripts.
Yeah, there is a. I already see there is another one on Arable Switch for you people.
Sure.
I mentioned, since the Arable Switch are conserved among bacteria, will it target and affect microbiota in human?
Yes, it will. That's a good question. Yeah, it will target, but I think many of these current antibiotic also targets the microbiota. So the alternative is to supplement the patients with probiotic drugs.
Mm-hmm.
Yep.
Okay.
Yeah.
I have another one for Fadi. It's also based in my. I don't know about Pseudomonas aeruginosa so much. It's I heard that there is some QS molecules in Pseudomonas aeruginosa. There is not only one. So the biosynthesis pathway is different among the different QS, or yeah, and you are blocking just one of these pathways, or?
Exactly. They're so interconnected, and there's 3 types of chemicals, and each scaffold or each chemical structure got their own synthetic pathway. So there's basically 3 sets of synthesis enzymes for different structures.
Okay. Great. I think that is more than time. It's 38. So thanks all the speakers for your outstanding presentations. I really appreciate it. Thanks a lot.
Thank you.
Thanks, Jordi.
Thank you very much.
Thank you very much, everyone.